So imagine you have this incredibly detailed, high-resolution blueprint of an enemy fortress.
Okay, I'm picturing it.
Right, so you know exactly where the walls are, you know the layout of the corridors, and you feel ready. But when you actually try to launch your attack, you realize the blueprint is, well, it's practically useless.
Because the defenses shift.
Exactly. The defenses shift, the corridors literally change shape, and your targeted strike just completely fails. And, that is the deeply frustrating reality of modern precision oncology for the vast majority of patients today.
It really is. We have this assumption that, that if we can just sequence a tumor's DNA, we get the blueprint, right? And then we can find the perfect drug to destroy it.
But today, we are doing a deep dive into a really fascinating 2025 paper from NPJ Precision Oncology. And their whole premise is that we might need to basically throw away that static blueprint entirely.
Yeah, it's a profound shift in how we approach the disease. Because the fundamental bottleneck we are facing right now is that genomics relying on that static DNA snapshot you mentioned, it really only provides actionable treatments for roughly, maybe 10% of patients.
10%, that's incredibly low.
Right. For the other 90%, the genetic mutations are just too complex or undefined, or they simply lack a corresponding targeted therapy. A tumor is not a static architectural structure. It is a highly dynamic, chaotic biological system.
So when DNA profiling hits a dead end for you, clinical oncologists often look to something called functional profiling.
Yes, the functional approach.
It's where you take a biopsy from the patient, isolate the cancer cells, and you grow them in a laboratory setting. We call these patient-derived cell cultures, or PDCs. And then you physically test different drugs on those living cells, right? You hit the fortress to see what makes it crumble.
You do, but that introduces an almost insurmountable scaling problem.
Right.
We have thousands of FDA-approved drugs available.
Yeah, and you can't test all of them.
Exactly. When a biopsy is extracted, the cellular material is incredibly finite. You physically cannot culture enough of a specific patient's tumor to test a library of, say, 4,000 compounds. It would be astronomically expensive.
And more importantly, a patient just does not have the time to wait for exhaustive month-long lab testing.
Time is the one thing they really don't have.
Which brings us to the core mission of this deep dive. The researchers behind this paper have engineered a mechanism to solve that exact scaling problem. They developed a methodology that uses a tiny biological taste test, combined with machine learning, to predict a tumor's specific vulnerabilities in just a matter of days.
It's incredibly clever. The architecture of their solution is built on a 4-step predictive framework. To start, step 1, you need a baseline.
Okay, the baseline.
Right. They compile a massive matrix of historical data. So, establish cell lines from past patients that have already been exposed to the entire exhaustive library of drugs. This gives us this vast, multidimensional map of bioactivity.
So we know how thousands of previous tumors reacted to thousands of drugs.
Exactly. Then, step 2 is where the actual clinical application begins for a new patient. Instead of wasting the precious biopsy material on thousands of tests, the clinicians apply a small probing panel.
The taste test.
The taste test, yeah. They expose the patient's cells to a highly curated selection of maybe 20 to 45 drugs.
And that probing panel, it generates a highly specific response vector for that individual patient.
Yes, which leads right into step 3. You feed that specific response vector, the bioactivity data, from just those 45 drugs into a machine learning algorithm. The algorithm basically maps the new patient's localized responses against that massive historical matrix we talked about.
So it's looking for patterns.
Right. And finally, step 4. The model extrapolates the data to predict how the new patient cells will react to the entire 1,000-strong drug library. And clinicians are handed a prioritized list of the top predicted therapeutic hits to actually validate.
I want to make sure we're fully visualizing how the algorithm makes that leap, because it really reminds me of the underlying math behind streaming service recommendation engines.
Oh, the Netflix analogy.
Yeah, but applied to cell death,
Yeah.
essentially. Like, if I watch 3 specific niche films, that's my probing panel, and I rate them highly, the algorithm maps my vector into its latent space.
It finds your cluster.
Right. It finds clusters of thousands of other users who had my exact reaction to those 3 films, analyzes their broader viewing habits, and predicts I will highly rate some specific documentary from a library of millions of movies.
That captures the relational logic perfectly. But we do need to refine the analogy just a bit to fit the biological reality here.
Okay, how so?
Well, the streaming algorithm is matching user preferences, which are highly subjective. In this paper, the methodology relies on transformational machine learning, or TML. It isn't just looking for an identical historical user, it is mapping the complex, nonlinear functional relationships between the drugs themselves.
Let's dig into that distinction. Because historically, when we talk about machine learning and drug discovery, we're usually talking about QSR models, right? Quantitative structure-activity relationships.
Yes, QSRR. That's been the standard for a long time.
And QSR is obsessed with the physical geometry of the drug molecule. Like, it assumes that if a molecule has a certain shape, it will bind to a certain protein.
Exactly. QSR models predict based on chemical structure, which requires us to assume we fully understand the drug's mechanism of action and the tumor's exact molecular target.
Which we often don't.
We very often don't. And that is why TML is so different. It bypasses that assumption entirely. By using the actual empirical bioactivity measurements as its fundamental feature set, it doesn't care why a drug works chemically. It only cares that it does work functionally.
It's a pure function over form.
Precisely. And to do this, the researchers primarily utilized random forest and gradient boosting regression models to map these functional relationships.
Now, a random forest model in this context, it isn't just making a single binary decision of, alive or dead.
Far from it. A random forest constructs dozens of distinct decision trees during the training phase. In this study, they defaulted to 50 trees.
50 distinct analytical pathways.
Yes. So imagine 50 different pathways simultaneously evaluating the cell viability scores. One tree might split the data based on how strongly the cells resisted drug A, while another tree looks at the subtle interaction between the resistance to drug B and the vulnerability to drug C.
That's incredibly detailed.
And then the model aggregates all 50 separate analyses to produce a highly stable prediction. That prevents the algorithm from overfitting to just some random quirk in the new patient's biopsy.
Okay, so let's do a quick checkpoint here. 5 minutes in, let's recap the core concepts for everyone listening.
Good idea.
Bullet 1. DNA profiling just isn't enough for cancer precision medicine anymore. We need functional testing. Bullet 2. Physically testing all drugs in a lab is way too expensive and requires too much tissue.
And bullet 3. TML uses a small drug probing panel, just a fraction of the library, to predict a tumor's reaction to thousands of other drugs.
Exactly. So I want to throw a question out to you, the listener. Imagine you only had 45 guesses to figure out an opponent's entire massive strategy. How accurate do you honestly think a machine could be with just that tiny fraction of data?
It's a daunting question, because the architecture of a model is really only as good as its predictive power in the real world.
Right, you can build a beautiful algorithm, but if it recommends the wrong oncology drugs, it's completely useless. And I was really struck by the sheer scale of the data sets they used to prove this concept.
You're talking about the PRISM dataset.
Yes, specifically PRISM. It contains over 500 patient cell lines tested against more than 4,500 FDA-approved drugs.
PRISM provides an incredibly rigorous testing ground. And the researchers set a particularly high bar for their model here by focusing on the prediction of what they call selective drugs.
Let's explain selective drugs, because predicting broad-spectrum drugs that kill everything is pretty easy, right?
Very easy. If you deploy a highly toxic, broad-spectrum chemotherapy, it will kill almost any cell line you apply it to. Predicting that is trivial.
So what's the real test?
The clinical value lies in identifying targeted therapies that are active in less than 20% of cell lines. These are the highly specialized compounds that destroy the specific tumor while sparing other healthy tissues. They're the needles in the haystack.
And the baseline probability of randomly finding one of those selective needles is remarkably low.
Very low.
When they ran the model against the GDSC1 dataset, which is another massive drug sensitivity database, the background hit rate for selective drugs was normally just 5%.
Meaning if a doctor were guessing blindly from a list of selective therapies, they would fail 95% of the time.
Right. But the performance of this TML recommender system starkly contrasts with that baseline.
It really does. When the algorithm was asked to provide its top 10 selective drug recommendations for a given cell line, an average of 4.3 of those recommendations were actual validated hits.
Wait, wow. 4.3 out of 10. That is a massive leap in efficiency.
It is.
You are moving from a 5% baseline probability to a scenario where nearly half of your top 10 recommendations are highly effective targeted therapies.
And the efficiency scales even more dramatically when applied to that massive 4,500 drug PRISM library we mentioned.
How so?
Well, the researchers constrained the model to a probing panel of only 45 drugs. That is roughly 1% of the total available library.
Just 1%.
Yes. And based solely on the tumor's reaction to that 1%, the model generated a prioritized list of its top 45 recommendations from the entire library. Consistently, those top recommendations contain between 25 and 38 highly active compounds.
So just by screening 1% of the library, they achieved 1,000 to 1,600% increase in hit rates compared to screening the whole library blindly.
The numbers are staggering.
And what makes this truly viable for wider clinical application is how much historical data it actually needs. You don't need millions of data points.
Nano.
Yeah.
The data decay curve they ran was highly encouraging. They systematically reduced the training cohort to observe when the predictive power would collapse.
And when did it?
Well, they found that an anchor of just 100 historical patient profiles retained the vast majority of the informational matrix required for accurate predictions.
Only 100 patients to train the algorithm. That drastically lowers the barrier to entry. I mean, a localized research hospital wouldn't need to depend on an impossibly large global database.
Exactly. A well-curated historical dataset of just 100 patients is sufficient. It makes the deployment of this technology highly practical.
All right, time for another quick 5-minute checkpoint summary. Let's recap segment 2.
Bullet 1. The model was tested on massive datasets, including up to 4,500 FDA-approved drugs.
Bullet 2. It completely excels at finding those elusive, selective drugs, which are notoriously hard to pinpoint and don't just poison everything.
And bullet 3, using just 1% of a drug library as a probe increases your hit rates by over 1,000%.
So we know the theory works beautifully on these pristine, established lab datasets. But here's the major friction point I kept returning to while reviewing the study.
The clinical reality.
Yeah, those datasets are immortalized cell lines that have been pampered in laboratory conditions for years. They are adapted to their environment. But what happens when you take a messy, chaotic, real biopsy from a sick patient today? Does the algorithm maintain its accuracy?
That is the ultimate question, and the researchers addressed it directly through their clinical validation phase, utilizing what they call the RX dataset.
The RX dataset. Tell us about that.
This was a bespoke dataset comprising fresh, primary tissue cultures derived from 24 individual patients representing various cancer types.
So these are real people currently fighting the disease.
Exactly. And the critical factor here is that the drug screens were executed immediately after the biopsies were extracted. They tested the fresh cells against a panel of 35 FDA-approved therapies.
Now, 24 patients is a relatively small sample size, so they couldn't just split the data in half for training and testing, right?
No, they couldn't. Instead, they utilized a statistical technique called leave one out cross-validation.
Okay, let's walk through that. To visualize this, imagine you have the 24 patient profiles. The algorithm intentionally blinds itself to patient number 1, trains itself on the complex relationships within the remaining 23 patients, and then attempts to predict the drug responses for the blinded patient.
And then it repeats that grueling process for every single patient individually.
Right. It is an incredibly rigorous validation method. It absolutely prevents the model from just relying on memorized data.
And to fully simulate the methodology, for every specific drug the model tried to predict, the remaining 34 drugs acted as the surrogate probing panel.
Okay, so let's look at the actual viability numbers, because this is where the clinical impact gets very real. When they exposed these fresh patient cells to the entire 35 drug library, the average cell viability remained incredibly high.
Around 87.5%.
87.5%. Most of the therapies applied did basically nothing to halt the progression of the cancer cells.
Which perfectly illustrates the complete inefficiency of a generalized approach.
Right. But when they isolated the top 5 drugs specifically recommended by the TML model for each individual patient,
the mean viability of the cancerous cells plummeted to 33.45%.
That is just a staggering reduction. You are taking a tumor that is essentially ignoring 87% of a drug library, and by trusting the algorithm's top 5 highly targeted selections, you drop its survival rate down to 1/3.
The study defined a definitive hit as any compound that suppressed cell viability below 30%. And across these messy, highly variable primary tissues, the methodology successfully identified nearly 69% of all available hits. If there was a highly toxic compound available in that library that could destroy the patient's specific tumor, The algorithm almost always placed it right there in its top recommendations.
And think about the timeline here, because time is the ultimate currency for an oncology patient.
Absolutely.
Culturing a massive amount of patient cells to physically screen a whole library takes way too long. But by requiring only a fraction of the drug library for that initial probing panel, this system can deliver a tailored selection of highly effective drugs within a week of the biopsy.
The speed is critical, but it also enables strategic combinatorial therapies.
Like drug cocktails.
Exactly. If a clinician knows exactly which 4 or 5 drugs are statistically most likely to induce cell death, they can formulate specialized cocktails immediately. They can preemptively outmaneuver the tumor before it even has the opportunity to develop acquired resistance.
Okay, so as we analyze the broader implications of all this, we have to talk about the data point that completely upended my understanding of oncology.
The tissue agnosticism.
Yes, the concept of tissue agnosticism. The traditional paradigm of oncology is fundamentally geographic.
It is. We categorize, study, and treat cancer based on its tissue of origin. Breast cancer, lung cancer, pancreatic cancer.
But the TML algorithm operates completely blind to that geographic origin. When the researchers analyzed which historical cell lines were providing the most informative data to predict a new tumor's vulnerabilities, they fully expected lung cancer cells to rely on historical lung cancer data.
But they didn't.
No. The algorithm frequently pulled the most critical predictive weights from entirely different organs. If you have lung cancer, the algorithm might determine that the functional vulnerabilities of your tumor perfectly mirror a historical bone cancer profile.
It reveals a really profound biological reality about convergent evolution in malignancies.
How do you mean?
Well, 2 distinct tumors originating in completely different tissues can independently mutate and stumble upon the exact same functional survival mechanisms.
That makes a lot of sense, actually.
And because the algorithm clusters these profiles based purely on how they react to the drugs, it identifies these functional overlaps. Now, the only notable exception observed in the data was with hematopoietic and lymphoid tissue cancers.
Blood cancers.
Right. They exhibited a strong preference for data derived from similar hematological malignancies. But for solid tumors, the boundaries blur.
That essentially introduces the concept of functional digital twins into precision medicine.
Digital twins, yes.
A tumor's identity isn't defined by where it started, but by how it behaves under chemical stress. You can map a new biopsy's response vector and find its identical functional twin in the historical database, even if one twin originated in the skin and the other in the liver.
It demands that we completely reconsider how we stratify patients in clinical trials. However, and this is crucial, it is equally important that we address the current limitations of this methodology.
Right, let's talk limitations. The researchers maintain a high degree of transparency here.
Yes, we have to acknowledge that this methodology is built entirely on in vitro data.
Meaning in the lab.
Exactly. The probing panel and the validation are occurring in a controlled cellular assay. And while the algorithm brilliantly captures the intrinsic vulnerabilities of the cancer cells themselves, an in vitro assay strips away the complex tumor microenvironment.
So it's missing the surrounding tissue.
It lacks the surrounding vasculature, the extracellular matrix, and crucially, it lacks the patient's immune cells.
Which means this specific methodology is currently blind to immunomodulators.
Precisely. Therapies designed not to poison the tumor directly, but to stimulate the patient's own immune system to recognize and attack the cancer.
Because the immune system isn't present in the little lab dish, the probing panel cannot capture that interaction.
Correct. The algorithm can only predict based on the features it can actually observe. Future iterations of this pipeline will likely need to incorporate more sophisticated organoid models that preserve a representation of the immune system.
Right. Okay, time for our final 5-minute checkpoint summary. Let's review segment 3.
Bullet 1. The model absolutely works on fresh, messy clinical biopsies, identifying highly toxic drugs for the tumor within just a week.
Bullet 2. The predictions are tissue agnostic. Breast cancer data can literally help cure lung cancer through the concept of digital twins.
And bullet 3. The model is currently limited when it comes to immune-based therapies because it operates in vitro.
So as we synthesize the scope of this entire deep dive, We are really witnessing a structural evolution in precision medicine.
We are.
We are moving away from the static, often unreadable DNA snapshot and embracing a dynamic, functional taste test.
By utilizing a highly concentrated probing panel and leveraging the pattern recognition capabilities of transformational machine learning, clinicians can save time, money, and potentially lives, completely bypassing the exhaustive trial and error phase of cancer treatment.
It is an elegant application of data science to biological complexity. It fundamentally changes the math of personalized treatment, but, it also leaves us with a really provocative question about the future of medicine itself.
What's that?
Well, if tumors are truly better understood by their functional drug response profiles, by their digital twins, rather than where they started in the body, are we approaching a future where tissue-based labels become completely obsolete?
That is a fascinating thought.
Think about it. Imagine walking into an oncology ward a decade from now, and you aren't separated into a breast cancer wing or a lung cancer wing. You aren't diagnosed based on the organ that failed.
You're diagnosed by function.
Yes, you are diagnosed as having functional profile type 42, grouped seamlessly with patients whose tumors share your exact biological vulnerabilities, regardless of where those tumors reside.
It would reframe hospital infrastructure, clinical trials, everything.
It's a massive paradigm shift, hiding inside a 45-drug algorithmic taste test. So think about how this changes our fundamental understanding of disease categories. Keep questioning the labels we've built, stay curious, and we will catch you on the next deep dive.